Good afternoon. My name is Juan Uriagareca. I'm an associate provost. And one of the reasons I'm here today is because I like lampreys and sea urchins very, very much. You see, I come from Galicia, and we actually eat them with lots of garlic, onion, parsley. It's delicious stuff. So through Avis Cohen's research, I've known now their significance for the nervous system in so-called higher vertebrates and their immune proteins. So I think I'm getting new understanding into the meaning of the concept of cannibalism. This is, I think, appropriate given Avis's integrative research spanning from mathematics to neurobiology with stops in mechanical engineering and cognitive psychology. And I don't mean so much because in interdisciplinary research, disciplines like those cannibalize one another, but rather because it is not uncommon for academic institutions to eat up anyone who seriously attempts such culinary feats. Now, the good news, I suppose, is that if you survive the process, you become distinguished teacher scholar, which is a rite of passage that we're here to celebrate today, together with Avis's 17th birthday. Birthday girl, from the bottom of my heart, I commend you for what you've done to lead an institution, ranging from NACS to advanced. You know how much I mean that. You've shown us the way, so thank you. And now to properly introduce you, another friend, Martha Nell Smith. She will seriously show you what she's all about. Thank you.
Well, I may or may not do that since I'm an English professor. I think about that. Can you all hear me?
Yes.
Okay. If I were in the audience, I might wonder why an English professor would be introducing this distinguished neuroscientist and all of the other things that Avis Cohen is. And to answer that question, I'll draw on one of her references for this well-deserved and long overdue award. And by the way, since she's an expert in several fields, Cohen's recommenders were all distinguished professors of engineering, mathematics, neuroinformatics, neuroscience, and organismal biology. Did I get that right? Yes. Okay. Okay. And anatomy, and from institutions you've never heard of, such as Chicago and Princeton. Anyway, one powerful letter of testimony acknowledges that she coaxed him out of a certain disciplinary blindness, and by doing so, helped to make his most important work imaginable and possible. She is one of the few who truly know how to reach across disciplinary boundaries, and is one who has been successful in teaching others how to do the same. Her admirers, and they are legion, go on to describe her truly revolutionary idea of applying computational models to explain locomotor rhythms and coupling, and her prescient understanding of the crucial importance of using the study of simple organisms and mathematical models and techniques to analyze the far more complex human organism. To declare this internationally esteemed, interdisciplinary, and I would say transdisciplinary scientist as being ahead of her time, as truly visionary, and her work as nothing short of landmark, none of her referees needed her numerous grants and awards, and we'd be here all afternoon if I listed those for you, nor her popularizing interviews with CNN, Wired, PC Magazine, and I could go on there too, but instead I'll point out that some guy named Einstein touted the importance of explaining the most difficult theoretical concepts so that they are legible enough for their significances to be understood by the general educated public. This English professor can definitely understand the importance of having been lead author on the paper that birthed computational neuroscience, of creating the Neuroscience and Cognoscience, or NACS as it's called, program upon her arrival here at Maryland. Cohen has long practiced the kind of interdisciplinary work widely regarded as key for our intellectual futures. Even more important, this intellectual leader of the highest order is committed to transferring knowledge and cultivating its production. In fact, Avis sees research itself as teaching. No wonder then that she is characterized by her students and colleagues alike in terms such as fabulous, intellectually stimulating, and incredible. Though I knew of Avis Cohen since she arrived on campus in 1990, and that was long before I met her, her stellar reputation is one that travels across the university as well as across the globe. I came to know and work with her directly during the years I chaired the University Library Council. I make mention of this because she proved a most impressive and valuable counselor, a distinguished citizen who keeps her promises, who volunteers for difficult and timely tasks when she recognizes their importance, and who can be counted on to deliver thorough, first-rate work and in a timely fashion. Obviously a team player and one who understands that the university cannot achieve the goals we have set for ourselves regarding excellence by remaining in our silos, Avis Cohen is a force for making dreams a reality, for confronting the most formidable challenges, and for solving seemingly intractable problems in order to make our world and its conditions conducive to growth for each and all. Now, I could go on, but you didn't come here to hear me. Yet I cannot close without telling you about one of her most important initiatives, which is again one that has brought people together, specifically women scholars from all over the campus. Avis Cohen is one of the co-PIs on the university's NSF Advanced Grant, aimed at increasing the recruitment and retention of women in academic, science, and engineering faculty. And not surprising for anything with which Avis is involved, the inclusivity of this high-impact award has gone well beyond women in STEM fields, science, technology, and mathematics, and targets the advancement and retention of all women faculty on our campus. Okay, I'm an English professor, and it won't surprise many of you to know that I cannot close without citing Emily Dickinson. And in this case, her words that the advanced program has taken as a motto, she wrote, "I took my power in my hands and went against the world." The most generous visionary, one who has made a profound difference on this campus, across this country, and around the world, Avis Cohen is truly a distinguished, and I mean distinguished, scholar teacher and mentor to us all. Rather than going against it, she takes the world along with her. And I will add, we are happy to follow. Please join me in honoring and welcoming Avis Cohen.
All I can say to that is that, like they say in Prairie Home Companion, if you need someone to write you a good letter, hire an English major. I could not have written such a letter. And I appreciate it very much. Thank you very much. And it is indeed a great show of feeling. I will say that we do have a very mixed audience, extremely mixed audience, ranging from my colleagues, my friends, my family, and they have training in just about everything, and some of them in not very much because they're still children. But nevertheless, I'm going to try to make it accessible to the lot. And therefore, if people have questions, I urge you to raise your hand and ask, because that's the only way that I can communicate. Is if I know that you don't understand me. So even if you're just lost, raise your hand and say, help. And I will back up and help. Okay? So, because I really do want this to be accessible. And so here we go. Okay. Now, let's see if I can do this. Okay, good. Now, I can't see. A journey with the Lamprey. How I learned I was an engineer. And I think I have learned that because I did find, if you want to come in, there's some seats in the front here. And you're walking, okay, you don't have to. And how I learned I was an engineer. Okay. It's a two-part journey that I'm going to talk about. The first part is the Lamprey, Spinal cord, and mathematics. And that will be the first part of this journey. And it was quite an interesting part. It's a very important part. The second part is motor principles that were obtained and taken to robots. And that's going to be the fun part, I think, for the kids, but and for me, because I also can call that playing with toys. And I do mean it because it is great fun. And where it all began was chatting with Phil Holmes over the Xerox machine in Cornell Math Department in 1980. Yes. It led to a collaboration with him and Richard Rand, and Philip as Further Books, and Richard as Further Books. And a collaboration started with Philip and actually has re-emerged in the last 10 years where we're working again together on these questions. So, now, if you want to read the Lamprey, here is one image of Lamprey mouth, but this is actually from the urban legend. So this is not quite what they look like. The real Lamprey looks like that. That's the mouth of a real Lamprey. So it's not quite so bad. You know, right, especially with gardeners. You know, they really aren't, you know, after all. The Lamprey is also known as 9 eyes in Europe and 8 eyes in Japan. And I don't know why the difference, if it's counting error or whatever. But it is a good vertebrate. Notice it doesn't have jaws, but it is a good vertebrate nevertheless. And it's at the bottom of the vertebrate tree. It's a basal vertebrate, to use the proper language. It's not a primitive vertebrate, it's a basal vertebrate. Bottom of the vertebrate tree. But it is a good vertebrate. And it is jawless, as you can clearly see. There's no jaw there. This is the tooth, by the way, that it drills a hole in its fish that it's clinging to and sucks. Like I would, but never. You know, everyone's got to make a living. They are jawless, as you can see, and basal. But the Lamprey nervous system has the same organization as higher vertebrates. It is possible, and I did this in 1988, to demonstrate a direct evolutionary path with controlled locomotion, which is what we're concerned about, from Lamprey to human. And in fact, it has all the parts of the nervous system that you have, not quite in the same proportions, but it has all the pieces. So the brain in all its parts is there and so on. So, and here's a whole lamprey. It has no appendages, as you can see. There's nothing on the side here. There's no appendages. It has these fins, and there's a lower fin also. There's no appendages. And everybody says, well, how big is it? That's, of course, a big question. And it swims, by the way, the traveling ways, which you'll see later. And they were also invited to attend the dinner. So, what can we say? Here's me holding one in a small tank. So you can see the size of a full adult. They're fairly big, but the ones that you get in the Great Lakes here are bigger. These ones are found in the rivers of Mississippi and so on. But the ones in Erie and so on are like this, that around, and they are big, and they are truly intimidating. Not like these guys are not so bad. These we can handle. The big ones, I don't know that I would want to do any experiments on them, but never mind. Okay. Now, the lamprey swimming, this is the lamprey starting to swim, and as you see, these are successive images. And to me, you can see that this traveling wave travels down the body. So it starts up here, and there's this wave here, and this wave travels down the tail, and this part is moving down and down and down and so on. And you have this traveling wave along the body, and if you record, I can stick little electrodes into the muscles of the lamprey very easily. Just little wires into the muscle, and they can record the activity of the muscles as the muscle is contracting. So it's an electrical signal that is generated from the muscle. There's some seats up here if anybody wants to come in. And that's what these are. These are the signals from these recording electrodes. And as you can see, one is going and two is going and notice that there is a delay from 1 to 2 to 2 and 1 and 3, that's these two here, are taking turns. They're alternating. Just like our legs, like this. That's what these are doing. They're taking turns. And the wave is traveling down in that way. So the traveling wave moves down the body, left and right sides alternate their activity, and the traveling wave travels uniformly. That is, it travels evenly along the body of the animal. So there's not much difference as it goes along the body, one part to the next. And the interesting thing is that this doesn't matter how fast the animal is going. Even if it's going very fast or very slow, you'll have the same traveling wave. And that is not an easy thing to accomplish, as you'll see. It's quite complex. And it's not, that is, how it does that is not truly understood. And in fact, lately I've had some new ideas about it might really be possible, but I will wait for that for later. I'm not going to talk about that here. Okay, so then this is, show constant phase relations among the segments. So the delay from here to here, here to here, here to here, is the same as it would be from here to there. So it's constant travel. Okay, this is from Ella Williams and Peter Valayan, but the interesting thing is that this pattern is exactly looks exactly the same as if you recorded it from an isolated spinal cord that's sitting in the dish. So I can take the spinal cord out of the animal, put it in a dish, record with the motor outputs, and it looks exactly, and I mean exactly like this. And there's some people in this audience who have recorded such things. As you will see here, this is taken from the experiments of Mick Mellon. And Mick is in the audience here. He did this very complex and it took a great deal of patience experiment. Working with Tim Keeble, they worked on, and Juan Lee is here too, that's right, Juan Lee was helping out with this as well. And you can see that here's the travel wave as it goes along the the body, and these are the outputs from, and this is an isolated spinal cord. This is not in the animal, this is in the dish. So it's really isolated, and this is the same activity that you just saw. So it really is exactly the same as in the isolated, it's in the whole animal and in the dish, okay? So it's not, if I were to take out a human spinal cord, don't worry, I'm not going to try. But, you know, if somebody were to do such a thing, the pattern would not be identical to what we do in life. But in this animal, the range of activities that the animal needs access to is not as great. So it doesn't have the complexity in its nervous system that we do. So it has the traveling wave in the spinal cord, and that's what comes out when you activate the spinal cord. Okay. So the isolated spinal cord can swim. We call that fictive swimming, which is like fictitious swimming, but it's the same thing. Okay. Now, if we look at a cross-section, so that is, take the animal and you go to, you make a cross-section of the animal's spinal cord, and you look at it, you will see that there are cells in here, and here, and there are fibers, axons here, and here, and they're coming in here, going out here. And then there's these big giant things, which are big giant fibers, that I'm not going to talk about at all. But this is the same basic organization of its spinal cord as ours. So you have a central part where the cells are located, then you have the outer part, which is where the fibers go, and then you have the descending stuff from the brain, the ascending stuff from the spinal cord, and so on. And it really is the same kind of organization that we have. It really is basically the same thing. This is looking down on it, and you can see it here. The other thing that people always ask is, well, how big is this? Well, I can tell you that the spinal cord, the width-wise, that is, let's see, what is it that it's about? Well, oh, that's right, it's about 100 millimeters. Avis. A millimeter wide, 250 microns. That's right. Okay. And it's small. It's small. You know, it's not so difficult to do. Okay. So, what preserves coordination? So, in that isolated spinal cord, you saw that there was this perfect traveling wave. What the hell is doing that? Hey, Nelson, if you want to sit there, sit here. Down here. Yeah, he's also got his isolated spinal cord. Right there. And even there, because I think that Charlotte's not coming back. So, what preserves it in that isolated spinal cord? Where's it coming from? The standard method in experiments with insect nervous system, was to take the nervous system out of the animal, put it in a dish, and make little cuts. Okay? You make a little cut, and you see, aha, I cut this, and now no longer we have coordination. It's all gone. That must have been where the coordination is. So that's the basic method that we're really looking at. Okay? We can make these lesions interrupt the coordination. Okay. So, looking at a cross-section of the spinal cord, there's fibers, going up and down, up and down, up and down. Long ones are out here. Long and short are in here. And this is where the cells are for the most part. So, if I cut off of the lateral tract, so I leave just a little bit here and here, this preserves the coordination. So, all you need is this, right? No. Because if I now do everything, I cut these as well, and just leave this, now you say, well, this is okay. So you can say, well, maybe it's just the fibers of the brain. It's just these connections. It's not the cells. You don't need the cells. You just need the connections. No, because now I can cut all the fibers into single cells and it helps nerves. So this is not an easy thing to figure out. Okay, this is going to be a difficult kind of study of the cake. What is going on here? This is non-trivial. Okay. So, no regions are necessary for coordination. They all can do it. Okay. That's pretty forward. All right. Now, summary of the lesions and different spinal cords. So spinal cords can differ. So for example, these guys, these black lines, they all destroy coordination. So some animals you need more than these ones do. Okay, well, fair enough. And here, for example, this one did not preserve coordination. So some animals are different. Now, why should that be? Well, we're going to see. We can come back to that. This is an interesting question that I can answer. I can't answer a whole lot of it, but this I can answer. Okay. So what? Here's the question. So what? What's the point? Okay. The neural network that generates this pattern is what we call the CPG, or the central pattern generator. It generates a pattern. It is the central pattern generator. Okay? So, what can we say about the structure of that network? The network has to generate the oscillations, these periodic, and it has to preserve that pattern. Okay. The order is getting taller. So, what can we say about the oscillator? This thing that makes those. Well, first of all, if we take an electrode and we stick it into a single cell, so here we got a single neuron, tiny little neuron, and some people have the ability, I'm not one of these, I have a little restless when it comes to sticking electrodes into neurons, but some people are very good at it. If you do that, you can actually, and then if you give TTX, TTX is the toxin, it's the poison that comes from one of the snakes, and it's one of the snakes that if it bites you, you're dead. But if you just take it out of a little bottle and you put it in a dish, what it does is it stops the neurons from talking to each other. They can't talk to each other anymore. They lose their ability to spike, so they use action sensors. They use sequences. But what you see is the periodic oscillation. Oh, that's interesting. So these cells are oscillating on their own. Well, okay. So we have the oscillator. That's the oscillator. Okay. And the segmental or unit oscillator consists of both induced oscillations and network oscillations. We know this from other things. But now, what else do we know about these oscillators? Do they have, for example, what's the frequency of the oscillator? How is that determined? That's a good question. Are the frequencies along the body, are they all the same? Are they all different? And if they're different, are they monotonic? Or are they just sort of random, all over the place? Well, that turns out to be a very interesting question that we were able to answer. And this result was predicted by Nancy Cattell, very distinguished mathematician from Taylor. And what she said is that the oscillations along the cord are going to be random. And I said, oh, Nancy, that can't be the case. There's order. There's got to be order in the universe. Some order anyway. And she said, nope, they're going to be random. The mathematician won the argument. They're not supposed to do that. They're not supposed to have the intuition that biologists have. Okay, so here's the evidence. Inclusion right. This is an animal where the head ends. So, what happened to that? Oh, that's right, here. So I took the cords and I cut them in half. Sorry about this, I forgot to explain. Cut them in half. And I got this piece and this piece, and I got the cordings here and here. now we can record from the head piece and the tail piece, and you get the head fast and the tail slow. Okay? That's quite a difference, that one. And here in this animal, the tail end is going faster than the head end. Okay? So they're not necessarily the right order. And here's one where they're roughly equal. So, and we did some where there were 3 pieces, and they really are random. There's no order to these. And in some cases, the frequencies are very different. In others, they're very much the same, as you can see here. So, how does it work? Okay. Well, first of all, I will just throw this out to people who are mathematically inclined and know some of the language. These oscillators are what are called limit cycle oscillators. That is, they're non-linear oscillators. They're stable to perturbation. You perturb them, they return to what they were doing before you perturb them. Okay. But we won't discuss that. It's just the math. Now, modeling in CPG is coupled oscillators. This is what I did with Richard before. It was later generalized by Nancy and Barbara McConnell. And I won't go into this, but it's a very simple set of equations just describing how the traveling wave, or how the phase delay changes from one moment to the next with one oscillator and another oscillator, and how they relate to each other if you have 2 oscillators and you have many oscillators. And it's just very simple. It has just that term of how rapidly things are changing their oscillation, and the frequency, and the coupling constant, and periodic function, and this is the base of the fusion function. Okay, that's it. Now, this is the equation, and this is the big equation. And that was the basic framework that Bill and Richard did. That was it. That was, at the time, it was basically revolutionary because it was so simple. And it has been framework for theoretical work and coupled oscillators for almost the last 30 years. It varied a lot, but that was basically the standard use. Okay. Now, the organization of the central pattern generator. So first of all, it's a distributed system. Okay, we've got the 2 oscillators, 2 per segment, and they're coupled across, and they're coupled up and down, long fibers and short fibers. And the oscillator is, as I said, nonlinear, blah, 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 capable of wide frequencies. And 10 key wounds also here. There. He's right there. Okay. Tim went on to show, and I'm not going to go into this at all because it's very complex, but he developed some very sophisticated mathematical methods for determining how strong the coupling was in the isolated cord. And that was very complex, beautiful mathematics that he did on that. And he was able to show that a lot of what we were doing. So whenever you make a model, and this is something about modeling that many people are aware of, whenever you make a model, you have assumptions. These are what you claim are true. You don't know that they're true, you're claiming they're true. And then you have the conclusions. Okay? And you disprove your assumptions. You don't disprove the model. You disprove that the assumptions of the model are incorrect. And then that model falls from the assumptions and so on. But all you're doing is, with that set of assumptions, this is what you can make a claim about. Nothing more. So that's what we did. We made a whole bunch of equations, a whole bunch of assumptions, some of them are true, some of them are false. One of them took 20 years to disprove. And we were thrilled. We finally disproved it. Hey! But that's actually what the whole point of the model is to set something you're trying to knock down. And it took us 20 years to do that. Okay, we did it. And there's more about the assumptions. But I think that some people are going to get a little bored. So I want to get into the engineering part. So welcome to Telluride. And on to engineers and Telluride. And who would ever say no to this? And in 1994, engineers wanted my input on neuromorphic engineering workshops. You might say, what is neuromorphic where we met. And neuromorphic engineering is engineering smart devices using the principles of neuroscience. They may not mimic biology, but they're going to embody the principles. That's what neuromorphic engineering is. And there are people in the audience who've been to that workshop, right? And probably some, I'm not sure. In any case, these principles have shown that they're important to make systems work. And it doesn't matter what the system is, whether it's made out of squishy stuff, metal stuff, or whatever. The system requires these principles. And these principles have evolved just as well as the anatomical components. So biologists talk about how evolution has shaped, all of the body parts, blah, 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 but it has also shaped the principle, how the components express those principles. And therefore, one can talk about the principles every bit as much as one can about the anatomical stuff. Okay, but how am I using it here? That's a fair question. What am I doing? I want to build smart devices that can control movement. Okay, that should be. I can see how animals move. Can I see how other creatures move? So, is it different from neuroscience? In motor systems, whether they're flesh or mechanical, the principles matter. The output is a product of its input. Okay, so you've got an input and you have to get an output. And that output has to be a function of the sensory inputs in some way or another. You have to know what has to be done and you have to do it appropriately. Okay. So, for example, the output, if I do something with my foot like this, it's going to be different than if I do something like that. That's what it means by reading the sensory input. Or if I'm leaning this way and trying to move, I'm going to do it differently than if I'm leaning that way. So, sensory inputs are shaped with the output is. That's critical. So, how has this worked? Okay, another fair question. You begin first with the simple aspects of spinal function. Just that first thing in the observation. We're going to start just with that, just to see what we can get from that. Okay, well, now, so this, by the way, is Ralph Asian Cummings, and is Shanique there? His wife and living boy were supposed to come. His Ralph was in Paris. So he's that's right. And this is the output of a chip that he designed, an analog VLSI chip. So most chips that people are aware of are digital chips. Chips in your computer are all digital chips. There are very few analog chips that are used commercially, but they're very low power and they're very efficient. And I'm not going to go through it all, but it's basically a capacitor that is charged up, blah, 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 and so on. Okay. The hardware implementation, this is how we laid out the chip, and it's all of 0.4 millimeters square. And it draws less than a microwatt power, so it's very tiny and uses very low power. Okay? So it's very efficient. It really is. And these little analog chips are very nice for that. Okay. Now, and it worked immediately. There's very few things in science that I can say work immediately. Very few. Exactly. Anyone who's done science, you know that the experiment does not work right away. This worked immediately. It really did. It was amazing. To control legs. And a passive knee. The robot was by Tony Lewis. That's Tony. And the sensors at the hip are right into the heart rate. And this is the robot, which I'll show you in a minute. And this is the control for that. Right? Well, it's just the legs. Sensory adaptation system. And here's one of them using the feedback. If you turn the feedback off, the light's going to go off in a minute. And And you see what happens when there's no sensory feedback. There it goes, light went off. Now it's having trouble. The hand of God comes and moves it back. And light comes back on, the sensors come back on, and it works again. Okay? So it needs the sensors. And there it goes. Okay, that's very nice. That's kind of cool. That looks like a nice little biped. Okay, and here it is. Running along, didn't you? One right, attack. The other one is what's called mirror control. And it's really doing pretty well. Oh, hey, great. Okay. Now, I'm not going to show you, I'm not going to go through this, but this is the two coupled limbs. And I have to emphasize that we put the control chip on this thing, and it worked within literally 5 minutes. This is, hang on just a second. Okay, this is the legs with no coupling. That's with unidirectional coupling. Right? Oh, come on. Stop. I didn't realize that it stopped to run it. That's not nice. My fault. Come on. All right, what am I doing? Okay, I'm just going to say it, but I'll put it in here quick. Okay, that's with no coupling. Unidirectional coupling. And bidirectional coupling. So you see, it really wants that bidirectional coupling. It really works very well that way. Okay. So, sorry about that. I just got some youngsters probably doing that. Okay, and then there's the other type. Okay. Here is that's Ralph. Used to run it when I worked with him. Case 28. This is
3, 2, 1.
and now they're coupled. Okay. 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 Oh, come on. Okay, now, what I'm going to do is turn on these different boxes. This one is the most fun. This is what happens when the sensor feedback isn't quite right. It does look a little drunk, doesn't it? I did not give it any alcohol before this. None. Okay. All right. Now, on to Japan and autonomous quadrupeds. So, all of this got the Japanese very interested in what I was doing because Japanese take their robots very seriously, as you probably know. They wanted to know how to control robots. And in particular, they wanted to know how to control legged robots. And I began to work with Hiroshi Kimura. Here's Hiroshi. I couldn't find a good picture of Yatsuhiro Fukuoka because she's too shy, but in any case. And Tekken. Tekken, I realized that Tekken has, I was told, I didn't realize, that it has meaning in Hebrew, which is very different, but in Japanese it means iron dog. So this is iron dog. Okay. It uses, this is how it's controlled. Read that. But here, Aichi Expo. And Aichi Expo was the world's fair in Japan. So, in the meantime, Yasser has gone on to his own position in Japan. He's now working on he's now working in his own lab. This creature here. It's loading up. But the interesting thing in this box is, you see, it's a biped. That's hard. That is very hard. I am really impressed. And they asked me to be a co-author on the paper because they thought it would give me credibility. I'm not going to say no. That's a great robot. So, we have some. But notice that it balances so well. It really works very well. Sarah, you're laughing. What? What? She's coming into this. I know. this one was more true to the central cabinet than it was really a very nice design. It's really done very well. You use that for stimulating mechanisms, and all kinds of work. So, I know, and this is a lamprey swimming in water with dye to show the vortices that gives off, because he's now used it to design a lamprey robot. Ah, yes, and we have Fan Sapien. We have, so for an encore, we have Mark Tilden, who is the designer of this guy, this girl, excuse me. This girl, this is Fan Sapien. Okay, here's Fan Sapien. This robot is sold commercially by Wally Toys, and I'm not making any money on this one. There's also a boy equivalent called Robo Sapien, and Mark, I met, and this is Robo Sapien, not quite that large. Mark was actually quite a large person, but he was very creative, and he was at Telluride, and was very interested in the stuff that I was teaching there, and he used the principles to build into his robots. This nice little bot, which we're going to demonstrate out in the hall, sells for under $100, which is pretty impressive because the robots that really work normally cost a lot of money, and he does it. I haven't seen her time safety before. He's done very nice at it. Thank you very much.